Spices have always been valued for their flavor and aroma, but aging science is now revealing something far more profound. Many spices contain bioactive compounds that interact directly with gene regulation, antioxidant pathways, and cellular repair systems that influence how quickly or slowly we age. Cinnamon, cloves, saffron, turmeric, ginger, rosemary, and cardamom all contain phytochemicals that modulate DNA methylation, histone activity, and stress-responsive signaling in ways that support longevity. What makes this area of research especially uplifting is how accessible these benefits are. Simply adding spices to everyday meals can create small but meaningful shifts in biological pathways that guard against molecular aging. Cinnamon offers one of the clearest examples. Its polyphenol and cinnamaldehyde influence insulin signaling pathways that regulate metabolic health, one of the strongest predictors of longevity. More importantly, cinnamon helps modulate transcription factors like NRF2, which turn on the body's own antioxidant genes. When these genes are activated, cells increase production of protective enzymes that neutralize free radicals and repair oxidative damage before it accumulates. Cinnamon also influences epigenetic enzymes involved in DNA methylation, helping stabilize gene expression patterns that tend to drift with age. By keeping metabolic and oxidative stress in check, cinnamon contributes to a cellular environment more aligned with youthful regulation and energy balance. Cloves, on the other hand, are among the richest natural sources of eugenol, a compound with remarkable anti-inflammatory and antioxidant potency. Eugenol helps regulate NF-kappa B, a transcription factor that, when overactivated, drives chronic inflammation and accelerates aging at the genetic level. By dampening NF-kappa B activity, cloves help turn down the inflammatory gene expression that contributes to cellular wear. Cloves also enhance glutathione activity, supporting deeper detoxification and reducing oxidative pressure on DNA. Their strong free radical quenching capacity improves redox balance in cells, helping maintain telomere stability and the integrity of mitochondrial DNA. These molecular effects mirror characteristics seen in slower aging tissues and populations. Saffron adds a uniquely positive dimension through its carotenoids, crocin, crocetanin, and safranal, which have powerful gene regulatory influence. These compounds modulate pathways related to stress resistance, particularly by enhancing mitochondrial efficiency and reducing oxidative burden. Saffron's bioactives support healthy neurotransmission, elevate brain-derived neurotrophic factor, BDNF, and calm neuroinflammation, all of which influence gene expression in neurons and glial cells. Early epigenetic studies show saffron can influence methylation of genes linked to mood regulation, memory, and inflammatory tone, suggesting that this spice helps maintain a more youthful neural environment. These effects help explain why saffron has such a strong historical reputation for supporting emotional and cognitive vitality. Several spices combine these benefits in synergistic ways. Curcumin from turmeric, for example, is well known for modulating histone acetylation and inhibiting inflammatory gene transcription. Ginger's gingerols and showgales help regulate oxidative stress and improve mitochondrial turnover. Rosemary's rosmarinic acid supports DNA repair enzymes and slows the accumulation of oxidative lesions. Cardamom influences metabolic gene expression and enhances natural detoxification pathways. Together, they form a broad-spectrum defense system that supports healthy gene expression far beyond what any single compound could accomplish. What makes spices especially promising is their ability to activate hormetic pathways, gentle stress response mechanisms that nudge cells into stronger repair, cleanup, and longevity modes. This is the same principle behind calorie restriction or exercise, but achieved in a small, sustainable way through dietary compounds. Many spice-derived phytochemicals mildly challenge cells in a way that boosts autophagy, enhances mitochondrial biogenesis, and promotes more efficient antioxidant activity. These responses help clear damaged molecules, rejuvenate cellular systems, and reduce the molecular drift associated with aging. Human research, though still evolving, is increasingly aligned with these mechanisms. Observational studies consistently show that diets rich in herbs and spices correlate with lower inflammation, better metabolic resilience, improved cognitive performance, and reduced age-related disease risk. Interventions with specific spices, cinnamon for glucose control, turmeric for inflammation, saffron for mood and cognition, cloves for glycemic and lipid balance, demonstrate real improvements in biomarkers tied to biological aging. Although epigenetic clock studies are still limited, early data suggests that spice consumption can modestly influence methylation patterns associated with lower inflammation and better metabolic regulation. Even more encouraging is how effortless incorporating spices can be. Adding cinnamon to oats or tea, using turmeric and ginger in soups or curries, including cloves in stews, or infusing saffron in rice or milk provides steady exposure to longevity-supportive compounds. Because spices are concentrated sources of bioactives, even small amounts contribute to gene regulatory benefits. Over years, these subtle shifts accumulate, helping maintain cellular stability and reduce the chronic stressors that push the body into accelerated aging. Spices won't freeze time, but they offer an appealing, natural, and scientifically grounded way to support the genetic and metabolic foundations of long-term health. Through epigenetic modulation, antioxidant enhancement, anti-inflammatory signaling, and improved metabolic balance, spices help create the conditions in which cells age more slowly and function more vibrantly. They transform everyday meals into gentle longevity interventions, flavorful, culturally rich, and deeply positive tools for supporting a more resilient, youthful biological trajectory.